All right.
Well, welcome
Ready?
everybody. I'd like to.
Oh, hold on.
Right.
Oh, okay, it is started.
Okay, started. Hi, I'm Wade Elmer, and I want to welcome everybody to our virtual seminar series. I think this is the second to the last one in the year, and I'm pleased to introduce someone from my department, Department of Plant Pathology and Ecology, Washington de Silva. If you don't know Washington, he's been here for about 18 months. I believe he arrived in the summer of 2019, and he is originally from Brazil, did his master's work at Louisiana, his PhD at Cornell, and then came directly to us, where he's been working on plant viruses. And one of his first assignments was to look at how plant viruses were affecting the grape industry here. It was an area that has just really not been examined well in the state. And so he's going to give a seminar on the survey for major grapevine viruses. And if you have a question, please put it into the chat box. And at the end of the seminar, I'll moderate the questions. Thank you.
All right, hello everyone. Thank you, Wade, for the nice introduction. Yeah, today we're gonna go viral. We're gonna talk about viruses and especially those that affect grapes. In this research in particular, we are focusing on three major diseases: grapevine leaf roll disease, the red blood disease, and also the thin leaf degeneration decline disease. I'll go over each one of them individually. The grapevine leaf roll disease is caused by 4 different species of viruses: grapevine leaf roll virus associated virus 1, 2, 3, and 4. The symptoms caused by this disease change depends on the type of variety of grapes. You have the red varieties. We see usually red leaves, as you can see here. If you take a closer look, it's actually very interesting looking symptoms in the leaves. In the white leaves, the white varieties, you see chlorosis, and sometimes you see also necrosis. What is common between them is that the leaves start growing. That's why the name of the disease, leaf roll disease. This disease also affects the grapes and deeply affecting the quality of the final product, either wine or juice, that whatever you're working with. I will touch base on this just a moment. So this is a picture I took last year from Rhode Island, a nice vineyard there in the state. And this plant right here in the middle was tested and was shown to be infected by leaf roll 1 and 3. And the plants on the side were all tested as well, and they're all negative. What was interesting about this picture right here is that once I removed those leaves in this location here and took a snapshot, we can clearly see the difference between an infected plant, grapes from infected plant, and grapes from a healthy plant. You see the healthy ones the berries are bigger, the clusters are bigger, and also the color is a little darker than the infected ones. And then this is more pictures from vineyards in Connecticut, and you see the same problem. And here we highlight here the uneven maturation of the berries, and as well as small clusters, abortion of the berries. So it's a big problem in the industry right now. Most of those virus species, the leaf roll viruses, are transmitted by vectors. I have three vectors here: the brown scale and cottony maple scale, and also the grape mealybug. All those vectors are found in Connecticut and New England in general. I absolutely like this picture because it shows 2 very dramatic change in the quality of the wine. This is wine made from healthy grapes, and this is grapes that are infected with GRV3, or leafy roll 3. You can clearly see that those infected by the virus have a very light color. And it is not just the color of the wine, it's also the quality of the wine. We have a reduction in soluble solids, and also the tannin and phenolic compounds. As Google says, this is pretty much a very bad quality vinegar, not even wine. So the point is, this really deeply affect the quality of the wine. The other disease is thin leaf degeneration decline. This is caused by more than 10 different species of viruses, and the major ones are grapevine thin leaf virus, tobacco, and ring spot virus as well. This is the grape leaf thin leaf virus. this is what the virus particle look like. And once we work in the lab and try to infect those things in the model plants, this is a bentamiana plant. We see all those very remarkable symptoms right here. What's remarkable about this virus is that vines that are infected by this virus tends to have the leaves change like a blade of a fan. That's why the fan leaf name, right? And you see here, this is a healthy one, this is an infected one. We see as well here, and then we see all this discoloration that's very common to the disease. We also see the shortening of the internodes of the plants, and over time the plants just collapse. This is a field in upstate New York and has been all taken by tobacco ring spot virus. This is a total loss, as you can see in this picture. And this is a picture showing a plant that's infected with tomato ring spot virus. And it's the same problem. We have a reduction in the size of the clusters, the berries, and also abortion of the berries. So deeply affect the yield and the quality of the final product in the grapes. Those viruses are called nipovirus because they are transmitted by nematodes. And this particular one, Rhizophyllum americano, it is present in Connecticut. And as I will show later, we have the virus or viruses, and now we have also the vector. So it's a widespread problem. And finally, the disease called Grapevine Red Blood disease is caused by Grapevine Red Blood virus. The symptoms are very close to what we see in leaf roll. We see a red discoloration or coloration of the red varieties of grapes, and the white ones we see chlorosis and necrosis in more advanced phase of the disease. The only thing is we often don't see the rolling of the leaves, so that might be the major difference between red blotch and leaf roll disease, right? And this is a DNA virus. All the other viruses I described before, they were all RNA virus. Red blotch has a huge impact in the quality of the grapes, maybe even more than leaf roll. As you see here, the grapes are all lighter than the healthy ones, right? And it doesn't take much to deduct of the quality of the wine that comes from those grapes, right? This virus has been shown to be transmitted by the three-cornered alfalfa tree hopper. Luckily, we don't have this insect in our region just yet. But I'm assuming with time, if the global warming happening, maybe they will reach up here. Okay, let's move on to our survey. The sample strategy that we use is called the stratified random quadrat distribution. And we see a plot right here, and we randomly assign quadrats to the plot, right? In each quadrat, we select 4 plants, and then we collect leaves samples from those plants. This represents 1 quadrat, 4 plants per quadrat, and then 4 leaves per plant. In total, we had 16 leaves per composite sample, right? And here are some of the students collecting samples from plants from one of the vineyards close by. And here more pictures about them doing the job. And we also try to collect insects, vectors of those viruses. We tag every single plant that we collect sample from, because later on, if you need to go back, we can track it back, and we did a couple of occasions, right? And the vectors that we found, we bring to the lab, we try to identify them, and then we test them to see if they have those viruses in them. Those samples are brought to the lab, and we process them. Here are the students happily processing the samples. We run techniques such as ELISA, RT-PCR, and PCR, and you do some sequencing to further analyze the data. And then the results so far, what we found is that in every single count in the state of Connecticut, we found leaf roll disease. For that matter, in every single vineyard visited. So in total so far, we visited 18 vineyards in Connecticut, Rhode Island, and Massachusetts. We collected 654 composite samples. Almost 10,000 single leaves were collected and processed. And we came to this, which is a little alarming. In each vineyard, over 50% of the samples were infected by at least one of the virus tested. This is very alarming. In some vineyards, the number went up to close to 100%. So the the problem is here, and it is widespread. That's what it's telling us. And then if you take a closer look, for all the samples analyzed, a little over 37% of the samples were infected by leaf roll associated virus 3, followed by leaf roll 2, 14%, leaf roll 1, 13%, and then tomato ring spot virus 10%, 10 leaf virus 8%, and then leaf roll 4, 6, and tobacco ring spot virus 5%. 37% of the samples tested were negative for all the viruses tested. And then just last week, this is the first time ever in this region, we detect 8 samples from Rhode Island. One of the vineyards that we visited there had a Grapevine Red Blood virus. So we are further analyze those samples to see if you can find more. And then we did some sequencing. Unfortunately, we had to stop the sequencing process because of the pandemic. Everything was shut down. We are going back right now, but we do have some data, and this is for GRV3. And based on this analysis we did, and we found that the isolates we found here are genetically diverse. As you can see, they cluster with strains from Portugal, from other locations in the US, and even locations from Brazil. This is a very early process analysis, but this is what is telling us right now. Hopefully in the next few months, we'll be able to collect more data and run some more analysis, have a better picture of what the diversity of all those viruses found here are in this location. So the question now is, what's the fuss? Why are we doing this, right? This is a picture of what is happening right now in Connecticut. As you see here, most of the plants are infected. There are displayed symptoms. People are losing money, right? Plants are dying right to left, and the growers just didn't know what was going on. And now we are starting revealing what the real cause of those deaths of the plants are. Here's more pictures, and always the same problem. We have uneven maturation of the grapes, which is a nightmare for the growers. They don't know when to harvest those plants. Those berries, they can be harvested too early or too late. The truth is there is not a good time to harvest when we have situations like this, uneven maturation of the grapes. In certain occasions, it's more concern is that even when the grapes are already ready for harvesting, the plants just die. What I have noticed is these plants are infected with those viruses when we have a shift in the climate here, in the weather in Connecticut, which is very common, especially when it gets too cold or too hot, the plants just collapse. And that seems to be a trend in the vineyards in our region. And then you have situations like this. This is, we had 18,000 plants planted here. It goes all the way up and down. And I took this picture right in the middle because more or less half of the plot was healthy. This is a 3 years old plantation. Those plants right here already bearing fruits, and those plants right here are suffering, right? And if you take a closer look, and that's what they look like. They have their leaves just rolling and discoloration. This is a Cabernet Franc variety. So we did some analysis, we collect a lot of samples here, and all those samples were infected by either leaf roll 1 or leaf roll 3, and sometimes a mix of them. So with that data in hand, we were able to help the grower to see what the problem was, and he ended up removing all those plants because this is a dead end, right? And according to him, this was almost $20,000 lost. And this is what you're talking about, people losing money, losing revenue due to this disease. And this seems to be a classic problem where what they say, right? If you plant a problem, you will harvest a problem. This seems to be the plant material is already infected, and that's what they are bringing to the vineyard. That's what they're doing, and you're trying to help them to have better strategy in planting those plants, and which I will touch base in just a moment. And then we have situations like this too. It's a small state, but we find all kinds of problems. This looks pretty much like a virus infection, right? Uneven maturation, the leaves are all turning yellow and chlorotic. And here another right, here you have more, and he's even more clear, right? So we test the samples and we couldn't find any viruses here. So we went ahead and did some extra tests, and the problem turned to be deficiency of boron and magnesium. And this goes to the say, right? Don't guess, get tested. Even for us, we try to call ourselves professionals. We try to say, you know what you're doing. And then when it comes to situations like this, it's not really easy cut. We really can't make any assumption. We have to test the plants to make sure that's the real problem, viruses. And then we came out to some other problems. Plants in several vineyards around here were dying left to right. They first developed some redness and then they start shrinking. And others, some that just collapse. We collect many samples from different plants, different locations, just different stage of growth. And we did a lot of analysis. We could not detect anything based on the test we were doing. So we select a few samples and decide to do a lunar high throughput sequencing. Right, and after much computational biology, we found a new virus to New England, which is called the Grapevine Red Stampeding Associated virus. This is not a new virus per se, because it's pretty much found in every grape-grown region, but it was the first time detected in this region. And we look at the literature, we see all those very clear symptoms where we have those galls being formed. And we look back, I have seen many plants in our region having the same problem. We at first thought was maybe crown gall because formation of the galls, but the difference here is that the galls are not very close to the soil line. It's more like in the upper part. So to me, it seems to be this virus causing the problems. And this is one case we test this plant. It died and was infected by the Rupestris stampeding virus. So now we are also testing for this virus to see if more plants and more vineyards are also having the same problem, which seems to be widespread. And then I select this case here, which is very interesting, right? Because that's why we do science. We find all kinds of stuff. Many growers, right, vineyards here in Connecticut and other states in New England, they grow other plants to be more like an attractive to customers to come and visit. And one of them is fig. And some growers are even trying to go commercial because there is a demand for figs in the region. But they are having serious problem with this disease right here. It's called fig mosaic disease. The leaves are the plants are in severe case, they just don't grow very well. And the fruits we see a lot of abortions, the fruit don't mature very well, and they just drop. We don't get a yield from those plants, right? So we end up testing those samples. We went to many different pharmacies, collect samples and tested. We did some lunar high throughput sequencing again. At this time, we used the plant ribosome RNA depletion assay, and we found two viruses. One is the fig mosaic virus, which is very much associated with fig mosaic disease. Right, and then we also find the fig virus 1, right? So that raises the question, what's really the culprit of the disease? And then we went through the literature. It's not really well defined. The only virus that they were able to fulfill partially Cox postulate is the FMV or fig mosaic virus. And the other viruses, they say, are associated with this disease. I think we have 11 viruses and 3 viroids associated with this disease. And it was quite interesting work. I had 2 brilliant students, Stephanie Praising. She was from Southern Connecticut. Now she's pursuing her PhD at Cornell. She started just last semester. And then the other one was Darlan Borges. He's from Brazil. He was here for 1 year in my lab. He's finishing up and he's defending his thesis next week. They did a wonderful job, a lot of analysis, especially genomic analysis, and they found a lot of problems in the genomics that are being deposited in NCBI database. And we put together this publication here, which is a very catchy name, a fig deal, a global look at the fig mosaic disease and its putative associates. And if you are interested, you can take a look. It's a feature article in plant disease. I just thought it was a very nice publication to put together because it really came from nowhere. So the question is now, you're doing all these, you're collecting all this data, so for what? What are you going to do with this? The major point here is really to help the growers. I always, at least twice a year, I haven't done this year because of the pandemic, we have grower meetings where I'm invited to give talks. I discuss my research, I try to raise awareness of the detrimental effects of plant viruses on the crops, Many people don't even know that plant viruses exist. And even though they are losing money, plants are dying, the production is down, the quality is not that great, right? So we are trying to come up with management strategies, and so far has been quite good. And then also we have meetings with policymakers. This is one meeting I had at the beginning of the year in Hartford. This is the Commission of Agriculture, where we really share ideas and try to bring up new techniques to solve the problems in agriculture. And this was all about viruses and all that, because in the future, where we are trying to head is to have a certification program, because now we detect the problem, we talk to the growers, we show them that all the plants are being pretty much infected, but then we don't know what to do because we don't have a solution. Once the plants are infected, this is for life, is a dead end. They have to just bear the consequences. And the major problem is that they are buying all those plant material, they're already infected by viruses, and they don't have other way to do it. And we look at those where they get those plants from. There is a tendency they all come from the same nursery close by, which is not even the state. So the ultimate plan is to be able to provide growers with a plant material that are test-free of viruses, right? So they have the option not to buy a little higher quality or better quality product to plant and not bring the problem to the vineyard and to the state of Connecticut and the region of New England. To give an idea why in Europe, a lot of vines are almost over 100 years old, sometimes even more. In California, over 50 years. In Connecticut, New England, the lifespan of those plants are less than 10 years. So every 4 years, 5 years, they have to replace all those plants. And they don't have where to buy healthy or test-free virus plants to plant. And that's a major problem we're trying to tackle right now, right? And with that, I would like to take a moment to thank all the people involved in this research and the other things in the lab too. My lab was really put together last year. The first time I would say it was functional was January 7, 2019. And so far we had a lot of people involved. And this brings me a lot of joy to be able to help to train the next generation of scientists. And we are doing that internationally. Here we have people from Brazil. We have a postdoc from China, 2 other students from Brazil, people doing a grad school already, others pursuing grad school, and that's really truly the light that we are looking for, which is really be able to work with these fantastic people. And we had produced a lot of results so far in less than 2 years. And I'm very motivated, and this is my motivation right here. And yeah, that's all I had for today. And thank you very, very much.
Thanks, Washington. Are there any questions? I'm looking in the chat box now, and the one down below, I'm not seeing any questions. But I have a few myself, if I can just ask those. I'm not too familiar again with these viruses again. The leaf roll virus, you've been out there for 2 seasons now. Do you see evidence of it spreading to asymptomatic fields yet?
No, I haven't, and it is just too early to say, to have that conclusion, because it takes up to 3 years to see any symptoms in the plants. As I show in that field, when they planted 3 years ago, and after going through all the process of taking care of the plants, that's when the symptoms appear. So if you have an infected plant, even though you might have the vectors that might be transmitting to new plants, it's not easy to see it. Even when you test it, sometimes you can get a false negative. Anyway, it takes 3 or 5 years to see any symptoms. Maybe if you ask me that question, maybe 3 years from now, I'll be able to have a better answer. It's just the biology of the virus.
Right. You say there's 4 types in that particular virus. Is that right? The leaf roll, there's 4 different types?
That's right. As far as I know, there is 4. It used to be 9 and then dropped to 7. And now I think the consensus that is 4. Because more analysis we do, we found that most of the species they consider single species are actually just a variation of the same species, right? And then, yeah, so now I think 4 is the right number to say, 4 species.
Do you think there's a reason for why the 3 is dominant here? I mean, do you think that was an initial contamination of the stock that originally came into the state? I think you mentioned that. Yeah, I'm sorry. And you mentioned in your survey that the leaf roll virus 3 comprised what, the majority of your samples this year?
Right. Yes. I believe that's the reason, because most of those infections, if not 100% of them, are coming from already infected plant material. But if you look at the distribution worldwide, not just in the state or in US, is that leaf roll 3 is the more predominant, the more prevalent virus when it comes to leaf roll disease. And leaf roll disease is safe to say is the most severe disease, the most important virus disease to grapes right now. I think red blotch is catching up, but leaf roll is still being the more detrimental disease.
Okay. My last question, it seems like this screams that we should maybe be thinking about establishing some kind of service for the grape industry so that we could start screening these products. You know, I know you and I have talked about this. What would you suggest would be the steps towards opening up a, you know, I don't remember the word, a validation of the tissue, yeah, screening.
Right, that's one of the goals too. Now it's nice because we have a lab that's functional and it can provide that service to the growers, right? We can test all the samples that they have. It comes with a cost. You know, it's not cheap to test plants or any virus sample right in the lab. I think we need to establish a process where the growers can actually pay for those services to be done, right? Because again, it's costly. But yes, it's very. I think that's where we should go, because unfortunately, having plants test free for viruses, that's not a reality right now, because we don't even have a nursery, as far as I know, that commercially sells plants for the growers. So they have to buy out of the state. And how can you implement a legislation that has some effect outside of the state, right? So I think being able to test those plants for them on site, that's for me is the best approach. It's just like we have to work around to see how we can do that, but it is definitely possible. Maybe it's the next step.
Okay, there are some messages above. Let's see. What will be the next steps for the newly found red blotch sample?
Yeah, that's a nice question. We just detected. We run a PCR. This is a DNA virus, right? And it was very, well, I should not say this, was very exciting when you find something new, right? So now we're gonna try to see see if other plants are infected too. We are trying to do some more studies, maybe some more sequencing on the isolates we have, and see if we can pinpoint where likely this isolate came from. Based on my guess, that will be from New York, because that's where they get the plants from. But anyway, the next step will be do some extra studies, do some sequencing and do some phylogenetic studies to see if you get some more information out of it.
Okay, I'm moving on up to the top of the list there. I was going in the wrong order. Here's one. How much do you observe co-isolation of multiple virus types on the same plant? Does being already infected by one virus make the plant more vulnerable to the other virus types?
That's a very interesting question. First, yes, we don't find it very often. I can't give you a number. I'll say maybe less than 5% of the samples had mixed infections. When you study virology, right, and from the notebook, when a plant's already infected by a virus, usually makes it harder for other viruses to come in. That's by by theory, but that doesn't seem to be really the case here. I think the plants, at least in Connecticut, for what I have seen, the plants becomes more, I wouldn't say susceptible, but weaker, and they're more susceptible to the effect of the weather rather than other pathogens from my experience. Viral pathogen, you know that fungal disease also take over when the plants are weak. For viruses, I don't know if that is a reality where you see uh, a plant infected by a virus become more susceptible to other infections. I think they all come together. I think, especially in our case, because even though we have the vectors, but they do such a good job in applying insecticides that we don't see much spread of the vectors spreading the virus. So to me, they all came already infected from the nursery.
Okay. We have a question here from someone saying that we have only one Concord grapevine, which we planted 45 years ago in our backyard. After the first 3 years, the grapes began to shrivel to raisins within a couple of weeks, leaves with brown, dry spots. The vine is still producing. How do we get that tested? I feel a bite.
Yeah. Well, it doesn't seem to be based on what you say. I don't believe it's a viral disease. It could be infected, but it seems like it's more like a fungal disease. We can send those samples to our clinic here in New Haven and see if we can detect something. But to me, based on what you're describing, I don't know. I would not say it is typical of a viral disease.
Okay, here one. How does the high incidence of virus infection here compare with California and Oregon? Are we worse off than they are?
I think based on what I am finding, we are much worse than they are. They do have big problems, but because they are so more technological advanced than we are in terms of production. They produce a lot of grapes, so they are on top of it, so they control it very quick. In our case, we are not. We are just starting. And for what I see, the incidence seems to be much worse than anywhere I have seen in the literature, statewide.
All right. Okay, we have a question here. We have 27 plus varieties of grapes here on the farm, I presume Lockwood Farm. I noticed the fruits of some varieties of grapes with thick skins did fine, while the others failed. Are some varieties more resistant to viruses than others? And if so, is it possible they could be crossed with more sensitive plants to enhance resistance?
Yeah, nice question. Depends on the virus you're talking about. As far as I know, there is not really natural resistance against any of those viruses. I might be wrong, but as far as I know, this is my best educated guess. So resistance is something you're not really working on. You more try to avoid to introduce the virus in your vineyard. For what you're describing, having a thick skin, I don't know, to me it's more like becomes more tolerant or resistant to other diseases caused by fungal or bacterial pathogens, not so much the viruses. And in terms of varieties are more resistant than others, there is this popular belief that the mixed varieties that we have in the hybrids that we have in US are more tolerant to viral infections. I think that's the belief that we have. But now the thing is, I see here a lot of those hybrids are 100% infected by the virus. I don't know if they show the same problems in terms of the yield should go down, the production should go down, the quality of the grapes should go down. I don't know much about it as far. There are some studies showing that yes, it happens the same way as the other varieties, the vinifera. But I think the vinifera are more susceptible in terms of showing the symptoms. In terms of acquiring the virus or the viruses, to me, it's just the same.
There's no more questions in the box that I see. I have one. I was curious about the sample you had where the boron and the magnesium deficiency was so prevalent. And I know that in Burgundy, France, where they grow really outstanding wine, the soils there are really high in calcium and magnesium. Do you think is there room for looking at how nutrition and how that could affect quality and also resistance? Because magnesium is really important in many host defense properties.
You talk about resistance to virus infection?
Resistance to a lot of things. I'm thinking primarily fungi, but I don't know about viruses. I just didn't know if you knew of anything about how nutrition. would affect
Well-
viral infections?
Well, for especially fungi, I think that software, especially, they use to control fungi. For viruses, I haven't seen anything out there. Anybody done any study, right? Clearly, that when the plants are deficient to some of those micronutrients, the grapes are just not good. They shrivel, they drop. The maturation is not even, just like virus infection per se. Yeah, I think there is a lot of room for studying and see those things. I know some folks are specialized in nutrition side, and yeah, I think there is much to do in terms of using nutrients to fight plant disease in general.
Okay, we have another question. We have only one Concord grapevine, which no, that was.
Yeah.
Is that the same question I saw earlier? After the first 3 years, the grapes begin to shrivel to raisins within a couple of weeks. Leaves. What can we do to cure it?
Oh, to cure it.
Oh,
Well,
that's
yeah, I think I don't. Yeah, maybe change a little bit. We need to know what the culprit is, right? If it is a fungal
Right.
disease, if it's a bacterial disease, and then we can come up with some management strategies. I'll just bring some samples to our clinic, and maybe they can, hopefully, they'll
Right.
be able to determine what's causing the disease.
That may have been a question I saw before. I'm not sure. Are there any other questions for Washington? We want him to start creating his own station brand of wine, so everybody here can taste it. You know.
Yeah, and also we are putting together a website, and hopefully soon. And when you have questions, we'll be able to guide you to the website, and we have a lot of nice information there. All the research you're doing on grapes, we're gonna post first-hand information there, so that the growers will have that as a resource to help them to make some changes.
I know there's a lot on your plate, but one last question I was curious about is, what do you think the reservoir of the virus is in surrounding weeds and other plants in the area? Have you begun to look at that as a reservoir for the viruses?
Well, I have not been looking at weeds per se. I did look at some wild varieties of grapes. You know, those are we see in the forest and close to the roads, especially close to the vineyards. And we have found a lot of positive ones, especially for leaf roll, which, I always say, oh, they apply a lot of insecticides, and there is a little chance that the virus is going to be spread from healthy ones to infected ones to healthy ones. But because you see those grapes from the wild grapes being infected, and there is a chance that that's being transmitted by the vector, being moved from the infected ones to the wild ones through a vector. But again, this is an early stage, and we still have a lot to do.
Yeah. Okay. I don't see any other questions, so let's give Washington a virtual clap. And thank you again.
Oh, thank you very much.
Thank you very much, Washington, for the wonderful presentation, and thank you, Wade, for the introduction and bringing a lot of good question to us. And thank you so much, and see you again next couple weeks. Thank you.
Okay.
Bye.
Bye-bye.